Symposium 2020. It's so lovely to have you join us today. I'd like to first acknowledge the first Australians on whose land we meet today and pay my respects to the elders past, present and emerging. And I live in Canberra, which is Ngunnawal country. So I'd like to welcome you today on behalf of the organising committee for the symposium. So we have Professor Lueck, Professor Lucas, Professor Cherbuin, Dr. Anne Bruestle, myself and Toby. So the whole committee would really like to welcome you today. So the symposium runs the whole week. So today's day one. So you'll have to be generous and forgiving as we get through the admin issues today. You're welcome to join us for each of the presentations. So today we have brain inflammation and aging with Professor Cherbuin and Dr. Bruestle. You have to register for each day separately, but we'd love to have you to join us if you can. So just a tiny bit of housekeeping. So if you can please leave your microphones on mute until the end of the session. We're going to have a Q&A session where you can ask the speakers questions about the presentations at the end. And you can also use the chat box if you want to add questions along the way, and I'll monitor the chat box as we go. This session is being recorded and it will be uploaded onto the website, so you can watch it again if you'd like to. The Q&A session, however, will not be recorded, so I'll stop the recording for that part of the session. So today's two speakers, I'd like to introduce you to them. So Dr. Anne Bruestle is going to present first, and she is the group leader in the Department of Immunology and Infectious Disease at the John Curtin School of Medical Research. Her work centres around the autoimmune aspects of multiple sclerosis, and she's the lead for the MS arm of the Our Health in Our Hands project at the ANU. And we also have Professor Nick Cherbuin. He is the head of the Centre of Research on Ageing, Health and Wellbeing at the Australian National University. He also leads up the neuroimaging and brain lab and oversees the neuroimaging arm of the longitudinal study called the Path Through Life study. His research interests include the risk and protective factors that contribute to premature aging, cognitive decline and dementia across the lifespan. So I'm going to stop sharing my screen so that Anne can share hers. And it's lovely to see all your faces and we welcome you all. And Anne, when you're ready, I'll get you to start. Yeah, I like the little wave there.
Hello, everybody. Thanks for joining us. And thanks for the introduction, Jo.
That's right. And we can see everything on your slides there. Is that okay? We can see your presenter view.
Oh. You can practice this as often as you want. It always happens when you try. What about this one? Is that better?
It's better. All good. Go, Anne.
Thank you. Okay. So I thought in the next 20 minutes, I would try to give a little bit of an overview of the blares in inflammation and the brain, to also set the stage a little bit for this week and give you the researchers' insight. And I would like to start with the structure of the brain. So I'm sure you all have seen these kind of images of the brain, and this is just a cross-sectional cut through them, and you've heard about gray matter and white matter. So what does that mean? So basically it just tells you about the composition of cells in these different structural components. And it is basically called white and gray matter just because when the first anatomes opened the brain, they saw white and gray, and that's where the names are coming from. So when we're looking at the gray matter, this is basically where the cell bodies of our neurons are sitting. So outside here are sitting, so this is my little comic of a neuron, are sitting the main bits of the neurons, while in the white matter, we have the axons. So the basically the information cables of the neurons, which connects them. And as you all know, myelin is the insulation around these axons. And because that's white, we call it white matter. We also have a bunch of other cells in the brain called glia cells, and I will go one-on-one on them in a little bit. So I'm sure you all have seen images like this, an MRI. So this is one way how we can actually see what is happening in the central nervous system. There are other ways to non-invasively look at the brain. For example, well, X-ray, then like imaging the vasculature, CTs, or PET scans, where we actually with a tracer can look for activity in the brain. So in research, we have a few more tools to do that. And of course, we don't normally have access to brain tissue, and we don't want that. But we have access to brain tissue of laboratory animals. And what we can do there is actually we can stain, we can visualize different structures of the brain. So here on the top is a staining which basically colors blue the myelin. So the white matter now becomes blue. And just as a fun fact, in the spinal cord, we have a reversion of this. So in the brain, we have the white matter inside and the cells outside, the gray matter outside, and in the spinal cord, it reverts. And our cells are inside and the myelin, the wires outside. We also can use different chemical reactions to emphasize certain structures or cells. And then we have ways to illuminate particular cells. For example, here, this is immunohistochemistry is the word for it. So we basically highlight a certain cell type we are interested in. So coming now to the cell types, I would like to start with this cell, which is an astrocyte. So astrocytes are all over your brain and spinal cord, and they are giving the brain the structure. They're also really important to nurture the neurons. So they're kind of the housekeepers of the brain. One astrocyte, and here's a picture of them, can interact with up to 2 million different synapses, so interactions between neurons. And you can see now why they're called astrocytes. It comes from the word star, astro. So here's a really, I thought, a really impressive picture of one. And one particular function of astrocytes is that they're building the blood-brain barrier. So blood-brain barrier, what does it actually mean? So basically, if you think of blood vascular as this like light pink tube and the blood cells inside, there's very tight connections between the cells. This is the endothelial cells of the blood. And then from the other side is the astrocytes, depicted here as well, building an additional layer. So, and this is really important because normally our immune cells circulate through the whole body and go into the tissue, while in the brain we have that very, very reduced. And that is why we call the brains an immune privileged site. So we don't have many of these infiltrating into the brain. So astrocytes are also important when we're now linking it back to MS, because given that there are the structural cells of the brain, they can actually lead to scars and basically preventing remyelination in lesions. So the next cell is the oligodendrocyte. And this is a cell which is really important in MS because these cells are the ones which actually make the myelin, which make the isolation, which is damaged in MS. And so it's really interesting. These cells can basically engulf or scaffold multiple different neurons at a time. And there are precursors of these cells, so renewable cells in the brain all the time. And this is one thing which actually is really highly important in the event of remyelination. And the last of the glia cells I would like to mention is the microglia. And these are the cell intrinsic immune cells of the brain intrinsic immune cells. So microglia are normally responsible for eating up debris and keeping the brain clean, the disposal of waste. But in certain situations, such as in MS, we can get these microglia activated, and then they can add to the destruction. So what does it mean, inflammation? And I would like to go back in history and tell you a little bit how inflammation is defined. So normally when we talk about inflammation, we're thinking of a wound, for example. And there's 5 pillars of inflammation, which are heat, redness, swelling, pain, and loss of function. So all of these factors are just a way to communicate to our body that something is not right, and to allow the immune cells to enter this area and to help, for example, fight bacteria or so which entered into the wound. And so here we come to the immune system, so the mediators of this inflammation. So as you know, our immune system is normally there to defend us against all different kinds of intruders, pathogens, bacteria, viruses, chemicals, any kind of insult. And we, as immunologists, divide them into 2 distinct populations. The one is called the innate immune system, which is really a broad functional unity of rapid responders. So part of the innate immune system would, for example, be physical barriers, your skin, if you think of this. But also certain cells which are there to react immediately against any kind of bacteria or virus. And then on the other side, we have this adaptive immune system, which is highly specific and targeted to, for example, a very specific kind of virus. But it also is able to launch immune reactions over and over again, and develops a kind of a memory of the intruder. So the innate system is broad, rapid, but not specific, while the adaptive is very specific, needs a little bit longer to get started, but is able to recap with the secondary infection. And so the mediators of this are soluble or cellular. And I'm going so much into detail here because it becomes important when we're thinking about what actually is inflammation in the brain. So the humoral, so the soluble factors of the innate immune systems are certain mediators. I'm sure you all heard about oxidative stress and the reactive oxidant species, so ROS. So this is what we all try to prevent with certain foods. But these are molecules which can be detrimental for harming other cells. Then there is certain molecules in the immune system called complement. These are produced by the liver, and they can, if they come to the, like for example, bind to bacteria, lyse these bacteria. And then we have all different kinds of other chemicals in the liquid phase of the immune system to help us defend us against bacteria and viruses. On the adaptive side, this part mainly defined by antibodies and cytokines, so hormones which with cells communicate between each other. And then we come to the cellular component, and I made another slide for that. So these are the main players of the immune system, which when things go wrong and our immune system attacks the own body, in the case of MS, the central nervous system, are there in the central nervous system and important for the pathology or the severity of the disease. So I talked about the microglia, so the cells which are normally in the brain and are part of the inflammation. Their equivalent in the bloodstream are macrophages. So what they're doing when they get activated is they're producing these reactive oxygen species. So they're producing a very inflammatory environment, which is normally important if you think of a bacteria entering a wound. but it's really bad if it happens in the wrong tissue, such as in the brain, where normally there is no cells. And then we have other players like natural killer cells and neutrophils, which I'm not going to go into today, but I would like to concentrate on the adaptive immune system. So I talked about antibodies. So antibodies are normally really positive because they again help us defend intruders, but when directed against the wrong thing, for example, the myelin, there can be doing harm. So these antibodies are produced by B cells. And you probably aware that many of the current treatments in MS are tackling exactly this the cell population. And then we have the T lymphocytes, so T cells. There are two different kinds. These green ones here are so-called cytotoxic T cells. So if they are activated, they produce molecules which can again harm other cells. They normally do that only when they see a cell which is not healthy, for example, a tumor cell or so. But in cases of autoimmunity, they can do that to our good cells, like the oligodendrocytes. And then are these CD4 T cells, which again produce hormones to attract other cells into the tissue. So that was a lot of biological background, I know. So I would like to bring it back into MS. So to do so, this is a review, so a review paper. I got this from Reich et al. It's 2 years old and was published in New England Journal of Medicine. So if you're really, really interested in the details around this, this is a really good read. So basically what Reich et al. Summarized there is when we're looking at active MS lesions, we see different types of immune infiltrations. And the most common one is where we see a lot of macrophages. These were the ones which produce the reactive oxygen species, and these T cells, which actually can damage other cells with their molecules they're secreting. And in a lesser extent, we see situations where in the lesions are also antibodies, as well as complement. So these other soluble components of the innate immune system. And the combination of this, complement and antibodies, can also damage cells, and in particular, the myelin. How exactly this is generated is still under investigation. And as I said, every person is different. So not every person has the same composition of immune cells leading to the lesions. So that also means that we need to know a bit more to inform what would be the best way forward. Yeah. So in the last 3 or 4 minutes, I would like to bring that all into the context of our current available treatments for MS. Because as you know, most of them are actually influencing the immune system. And what's very interesting is, even though we know that the damage is done by the cells in the central nervous system, in the brain and the spinal cord, most of our treatments are actually in the periphery. So all they do is either reduce the potential of cells which could do harm by just suppressing the immune system, or taking out certain players, or shifting the immune system from a more inflammatory into a more anti-inflammatory situation. So there's a few examples of cells which are actually, or like of treatments which are actually targeted a little bit more into the functional brain inflammation. So one of them is natalizumab. So natalizumab is basically blocking these T and B cells from entering into the brain. So here we have, again, the blood vessel and our endothelial cells, and here the astrocytes building the other side of the blood-brain barrier. And what happens in MS is that these cells, the T cells and the B cells, go inside and do their damage. And natalizumab inhibits this. Ocrelizumab, again, is an depletes in particular the B cells, and reduces the effects in that way. And then when we look into a lesion, there is other treatments which seem to happen there. Some are affecting functional properties, while other like newer treatments are trying to induce neuroprotection rather than just suppressing the inflammation from the outskirts. And so I think it's a good summary slide because you can see all the players again. So we have the oligodendrocytes, which wrap the neurons. We have the astrocytes, which building the blood-brain barrier. We have the microglia, which can normally scoop up debris, but can also be very detrimental to our oligodendrocytes. We have the oligodendrocyte precursor cells, which I mentioned briefly, which can come in and lead to a remyelination of a damaged myelin sheet. And then we have our infiltrating bad guys, the T cells here, complement. And I don't think they depicted antibodies, but that would be from the B cells. Yeah, so what can we do as immunological researchers to actually help develop our understanding of what's happening in inflammation in the brain? So what, for example, my team does is we're using a animal model to model these effects I just described. These effects which lead to the demyelation. And how it works is, as you know, and you will hear a lot in this week, MS is a very personal affair. So everybody of us has different compositions, what actually led to the development of MS. So, and these factors can be enough to develop MS for one person, but not for another. And so it makes it really, really complex. But what we know is whatever the composition was, we have these immune cells I was just talking about, like the T cells and the macrophages going into the brain and doing the damage. So instead of trying to figure out what's the composition in a particular person, we're using an animal model where we directly activate the immune system and causing the same thing. So by now manipulating these animals and taking out certain cells or taking out actions in these cells, we can investigate how these cells or their function are involved in the development. And only when we know enough of this, we can bring forward our treatment options. Yeah, and with that, I would like to hand over to Nick, who will talk a little bit more about inflammation in the context of aging.
Thank you very much, Anne. I'm also going to share my screen. Can you see my slides? Yes? Very well. Well, welcome everybody, to this MS 2020 Symposium. We're very glad to have you here. Now that Anne has clarified the details of the inflammatory response in MS, in this second part of the presentation, we will discuss how inflammation impacts our body in a more general way, how it's related to the aging process, our lifestyle, and the development of chronic disease, and why it matters for our brain. This is important because MS-related inflammation occurs not in isolation, but in addition to typical inflammation processes that we all experience to different degrees throughout our lives. So a brief overview, we'll look at how aging affects our brain, what are some of the main risk factors for brain aging, why do we age, how is aging related to inflammation, how does inflammation impact our brain, and what contributes to inflammation, and finally, what we can do about it. But before we get to this, I would like you to think about this presentation as a glass half full. discussions about aging tend to highlight how things go downhill with time and can be a little depressing, to be frank. So while there'll be some of this in this talk, if there's a single message I would like you to come away with, is that we can do something about it and have a positive influence on how we age and how we manage inflammation. So you should see what comes next in this light, in the, you know, the bit we can do something about. So aging, we often think of aging as something that occurs later in life and which is characterized by wrinkles, frailty, and as is clearly illustrated in this picture, difficulty in mobility and in thinking rationally. However, these are consequences and signs of aging. The aging process actually starts much earlier at conception. So why do we age? Well, aging can be broadly thought of as a combination of 4 types of biological processes and the progressive breakdown. From conception, our cells' genetic materials are constantly being damaged by things in our internal and external environment, such as viruses, injuries, inflammation, UV exposure, etc. Fortunately, our cells have developed very effective repair mechanisms which can fix damage as it occurs, although not perfectly. Another contributor to aging is manufacturing problems. Our cells are very good at producing all sorts of proteins and substances necessary for normal function, but sometimes they get it wrong and it creates unwanted rubbish. Again, fortunately, we have also evolved excellent rubbish removal mechanisms to avoid our systems becoming clogged up with rubbish. But typically, a little rubbish is left behind. So because repair and rubbish removal mechanisms are not perfect, with time progressively more and more damage and rubbish accumulates, which in turn further impairs maintenance, and this leads to a progressively accelerating aging process, which becomes more and more apparent in midlife and onwards. But we have to keep in mind that this really occurs throughout the lifespan. It doesn't start suddenly in adulthood or later. So at a very high level, what does brain aging look like? We can see on this slide, on the left, a graph showing the shrinkage of the brain between the ages of 80, or the shrinkage of the gray matter between the ages of 20 and 80. And on the right-hand side, the shrinkage of the white matter, which contains the axons and the myelin attacked in MS. And we can see that for gray matter, there is a fairly consistent decrease in volume between the ages of 20 and 80, while the white matter increases somewhat until midlife and then decreases at an accelerating rate. And to give you an idea of how big these changes are, we lose about 13% of our gray matter across our lifetime, and about 25% of our white matter, with an average of about 20% for the whole brain. But of course, this is very different in different individuals. So it can be much more in dementia, for instance, or it can be much lower in people who have particularly good lifestyles and also who are lucky to have maybe better or healthier genes, which we can't control. So how are these volume changes reflected in the structure of our brain? We might think that it's mostly about losing neurons, since when we think about the brain, we think about neurons. But actually, we lose only about 10% of our neurons over our lifespan, and they're relatively small cells. So in fact, a lot of the loss of volume is due to the loss of cell components and damage to cell membrane. We can see here on the right-hand side of the slide, first a healthy neuron that has more branches, and you can see it's got along the branches, there are many of these little things poking out, and which connect to other neurons. In an older, less healthy neuron, there are less branches and there are less of these connections as well. Another important component of volume loss in aging is the loss of glial cells. We could lose as many as 26% of them across the lifespan, and explain how some of them contribute to the maintenance of the brain, but also the repair of the myelin sheath around the axons. So that's very important. Now, I can only talk about a few of these changes. Obviously, there are many more things that happen in the brain, like decreased production of neurons and other things. Of course, these changes also lead to changes in function. And so across the lifespan here, we can see changes in different cognitive domains from age 20 to 80. And we can see that in many domains, we see a very consistent decrease in cognitive function, but not all of them. So there are some that remain relatively stable and some that even increase, like work knowledge, vocabulary, and there is a lot of research around wisdom as well. So what are some of the main modifiable risk factors that are linked to brain aging and cognitive aging? So on this slide, I've selected a number of risk factors that are very well established. The research, it's not just about one single study showing that this or that is a risk. It's based on very large studies and meta-analysis that have surveyed tens of thousands of individuals. And so we're quite confident about these effects. And some of the major risk factors are smoking, obesity, anxiety and depression, excessive alcohol intake, cardiovascular disease, an unhealthy diet, type 2 diabetes, an inactive lifestyle, poor sleep quality, high cholesterol, and social isolation. You'll note that many of these risk factors are risk factors for cardiovascular disease. And this should not be a surprise, because as you can see in this image, this shows all the blood vessels in the brain when the tissue is removed. And there are about 600 kilometers of capillaries and blood vessels across the brain. So you can imagine that if with increased cardiovascular disease, if these get clogged up, it will lead to major brain dysfunctions. So how is inflammation that occurs throughout our body linked to brain aging? Anne alluded to this. One of the most potent biological mechanisms implicated in biological aging, and which explains a substantial part of the brain aging effects we have discussed in the previous slides, is called oxidative stress. In order to survive, our cells need to produce energy. This is achieved by an oxidative process that combines the oxygen we breathe with other molecules to produce energy. However, these processes also produce byproducts called reactive oxygen species, or ROS, and these molecules are able to react with components of our cells, and in the process they damage DNA, cell membranes, and other cellular structures. Now, fortunately, our body has defense against reactive oxygen species, produce antioxidants molecules, or it acquires them from our environment through our diet, which neutralizes the reactive oxygen species. So the balance between these reactive oxygen species that are being produced through oxidation and those neutralized by antioxidant determines the amount of oxidative stress that is present in the body and how much damage is produced. In practice, our cells are continuously under some oxidative stress, but it varies. And this produces higher or lower levels of inflammation in our body and leads to cellular aging. As we've discussed before, our cells have several mechanisms to fix damage caused by oxidative stress and other biological processes. But since they're not perfect, damage accumulates. And one issue as well is that oxidative stress increases and activates inflammation, and inflammatory processes also activate oxidative stress. So they sort of self increase one another. Importantly, in the brain, this leads, this combination of oxidative stress and inflammation leads to decreased neurogenesis, so decreased production of new neurons. It leads to an increased misfolding of protein and the accumulation in the brain in plaques, such as in Alzheimer's disease. It leads to an increased risk of stroke and mini strokes, and an increased shrinkage of the brain as well. And importantly, as inflammation increases, it increases the risk of developing certain chronic diseases, such as cardiovascular disease and others that are presented here in this slide. You might, I'm just going to point to one. You might be surprised to see that inflammation might lead to a problem with diet. One would rather think that it goes the other way, is that poor diet leads to inflammation. And as we'll see in a second, this is indeed the case. But new research shows that inflammation can affect certain brain centers and damage them, and lead us to overconsume food, which then leads to more oxidative stress and more inflammation. And importantly, many of these these risk factors and others lead to increased inflammation. So again, we've got a vicious circle of inflammation leading to chronic disease leading to increased inflammation. But the good news is we can do something about it. The exercise, for instance, is known. Exercise, maintaining healthy weight or decreasing body weight, and a healthy diet are known to both lead to the production of the increased production of antioxidants and the downregulation, the suppressing of inflammatory processes. There is also emerging evidence that stress reduction, which is a bit harder to study in this context, and social connectedness and decreased depression and anxiety also lead to decreased inflammation and a healthier brain. Now, I'd like to now is also note this research, which has been done in a tribe in Bolivia, the Tsimane tribe, because we might think, well, because we're talking about inflammation in our body related to health and lifestyle, and inflammation linked to MS, which we might not be able to suppress to the same degree. How much can we influence the information we have in our body? And what's interesting about this research is that the researchers have looked at the amount of cardiovascular, the quality of the cardiovascular disease that develops in this tribe. So this is in the dotted line in blue is the Tsimane tribe across the ages of 40 to 84. And we can see that while the the healthy cardiovascular health decreases a bit in the Tsimane, there are relatively few who develop a high level, a very bad cardiovascular disease. Whereas in Western cultures, the increase in cardiovascular disease of the worst kind becomes extremely prevalent at all the ages, 50%, and those who remain very healthy with no sign of cardiovascular disease decreases to a quite low level. And what is interesting about this research is that it's been found that in the Tsimane tribe, the inflammation levels actually are quite high. They're higher than in Western cultures. So it is possible and what characterizes this tribe is that they've got a very healthy lifestyle. They exercise a lot. That's part of the daily activities. They mostly use a plant and fruit-based diet with relatively little meat, and they're very social. So it's possible to have a relatively high level of inflammation, which presumably in this tribe is somewhat buffered by their lifestyle. And to finish, I want to point to Our Health in Our Hands. That's an ANU initiative that aims to develop research to improve the treatment and the understanding of disease, and particularly MS and type 2 diabetes. And the reason I want to talk about this is because they're very important projects, and it relates to what we've discussed in today's talk. And that is that some inflammation comes from the MS pathological processes, the MS disease, and other from our lifestyle and what happens normally in the body, and they come together. And it can be difficult to separate the two in MS, and to really understand what contributes. Is it aging that has these effects in MS, or is it the disease itself? So what we aim to do in Our Health in Our Hands is to develop an MS cohort that we will follow over time to start to answer these questions. And what's special about this cohort study that is not often found in MS research is that we're going to recruit a non-MS control group as well, so that we can start measuring and understanding how much lifestyle contributes to the progression or non-progression of the disease. And I can't give you much more details now because we're running out of time, but there'll be a link on the ANU website, and you'll hear more information throughout the week about this research as well. But just for now, this is what it will look like. We will follow people over time with annual follow-ups, and this will give us the opportunity to detect relapses and then relate them to lifestyle or other disease exposures. And for this, we need your help, especially if you live in the Canberra region. So I've put a link to this research. It's just been put up on the ANU website because we've received the ethics approval to conduct this research. So you can just either use the link that is provided on this slide, or search ANU Ohio and MS research, and you'll land on it. Thank you very much for your attention.